Good morning, good afternoon, and good evening, everyone, and welcome to this Scientist.com webinar titled Spatial Phenotyping, a Revolutionary Approach to Biomarker Discovery for Cancer Immunotherapy. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host today. This webinar has been sponsored by Akoya Biosciences, so big thanks to them for helping to make this webinar possible. We're very fortunate to be joined by Dr. Arutha Kulasinghe, Senior Research Fellow and leader of the Clinical Omics Lab at the University of Queensland and founding Scientific Director of the Queensland Spatial Biology Centre. Today, Dr. Kulasinghe will discuss how single-cell spatial phenotyping is an invaluable tool when characterizing the tumor microenvironment and how this technology can be used to develop consistently predictive biomarkers for guiding patient selection for highly targeted therapy. And with that, I'd like to welcome Dr. Arutha Kulasinghe. Arutha, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you for the invitation to speak today to Scientist.com and Akoya Biosciences. Today I'll be talking to you about from pixels to clinical insights, a really deep dive on the tumor microenvironment in lung cancer, head and neck cancer, and skin cancers. My name is Arutha Kulasinghe. I'm from the Clinical Omics Lab, a lab which I lead at the Fraser Institute at the University of Queensland in sunny Brisbane, Australia. I'd like to acknowledge a number of funding agencies that support this work, both nationally and internationally. And with that, I think we'll probably kick off. So immunotherapies are sort of the mainstay of cancer therapeutics at the moment, since all of Jim Allison's work in CTLA-4, and then more recently PD-1 targeted therapy, PD-L1, PD-1 targeted therapies. These therapies have revolutionized the field of solid cancer treatment. The work's really led in the field by what's been done in melanoma, non-small cell lung cancer, head and neck, bladder, and so on. Even in triple-negative breast cancer, we're seeing a subset of patients that appear to do well. One of the challenges with these therapies is that they cost $150,000, $200,000 per patient per year. So these therapies cannot be given blindly. They only work in about 15 to 30% of patients. And so one of the unmet clinical needs at the moment is how do we identify these patients that will benefit from immune checkpoint inhibitor therapy? How do we identify that 15 to 30% of patients? And one of the real pivot sort of points is how do we identify these patients? But also there are patients that receive immune-related adverse effects. So how do you identify those patients that'll, potentially not benefit from these therapies or potentially need combination therapies to really target the individualized tumor? And so this really poignant piece came out in Nature, a commentary a couple of years ago. And it was a really interesting piece, and it showed that tissues and not blood are where immune cells function. And it was a really nice piece showcasing why we need to capture the proteomic data, the transcriptomic data, but simultaneously capture the imaging data as well. The imaging data is just as important as all the molecular measurements, and putting those data sets together and being able to see that on a slide, almost this digital pathology revolution, is really powerful because we can start to integrate and understand these data sets together. I did my PhD in the liquid biopsy space, and when I was doing that, it really reminded me of why we really needed to go back into the tumor and potentially metastatic tissue as well, and really try to understand these cell by cell. And so there's a growing need to improve immune prediction of response. There are a number of predictive biomarkers in the space: PD-1 expression, PD-L1, tumor mutation burden. These do appear to be sort of limited in their scope. So TMB is a sort of a bulk neoantigen load measurement. You can look at the number of mutations in a tumor. High tumor burden sort of signals high neoantigen load, and therefore the immune responses are greater, or that's what the sort of common thinking is. PD-L1 expression in the tissue, high expression generally means that you can give these patients these anti-PD-1, PD-L1 therapies. However, we do know that in patients with low and no PD-L1 expression, some do receive benefit. So when you look at the MSK impact studies and some of those sort of really landmark studies, we can see that these predictive biomarkers appear to identify independent groups of patients that will respond to therapy. So it's not a sort of a holistic biomarker in that it doesn't really capture the dynamic state of the tumor. and the tumor microenvironment. And so we know that the tumor microenvironment is made up of millions of individual cells. This is sort of this milieu of tumor cells, immune cells, dendritic cells, endothelium, vasculature, all these sort of really interesting cell types, B cells, TLSs, and so on. And what my lab's trying to really do at the moment is try to do a deep dive on this tumor microenvironment, because we know that, when we talk about heterogeneity and things like that, we know that tumors are very heterogeneous, but can we visualize them? Can we understand them? Can we identify areas of sensitivity and resistance? And this is sort of that deep dive into the TME that my lab's been able to do. And I'll talk about some of those studies in this talk today. And so why this is important is if we look at these two tumors at the bottom left here, we can see that the tumor, if we were to grind these two tumors up and look at a bulk expression score, so a bulk tumor infiltrating score, we can see that clearly on the tumor on the left doesn't have as high an infiltration with the sort of purple cells infiltrating the tumor as the tumor on the right. So we know at a bulk expression level, these are very similar tumors, so a similar TIS score, a similar tumor infiltrating score, but the tumor on the right is highly infiltrated compared to the one on the left. And when we know that a highly infiltrated tumor where the immune cells are activated tend to do better with immune checkpoint inhibitor therapies, whereas if we were to just grind these two tumors up, we'd get very similar scores and potentially not be able to differentiate the two tumors. And so there's a growing need to understand tissue complexity. Back in the day, we did bulk genomics, where you're looking at average expression counts across different markers, the fruit salad analogy, which has probably been overused, but it's a great example of how this field really revolutionized and evolved over the years. And in 2011, with Nick Navin's work, we went on to see the development of single cell sequencing, where you could sort of move on from this fruit smoothie, where you have averaged expression counts for different markers, to looking at the different proportions of the individual cells that are being composed in these tumors. So you can look at the individual blueberries, the strawberries, and so on, analogous to tumors. And so what's happened in the last couple of years is we've moved into this spatial transcriptomics, spatial proteomics space, where we're able to profile the entire tumor without having to dissociate or mash these tissues up. We can see the disease for where it is in the body, we can see it on the organ itself, and we can see how those individual cells are not only composed, how many are there, but where are they, how are they talking to each other, what are the neighborhoods, what are the cellular communications that are going on. And that becomes really powerful in trying to understand how the organ is adapting to these tumors that are growing in them. How are the immune cells that are infiltrating the tumor? Are they on the periphery? Are they in fact infiltrating? And it's not just composition, but it's location, and getting all that sort of tissue contexture is really important. And so this is what the revolution's been over the last couple of years. And there was the Nature Method of the Year. So it's giving us deep insights into new biology. We've written a couple of pieces in GenEng Biotech, top 10 spatial biology companies, how spatial biology is redefining the multi-omics approach, how it's reorienting biopharma. And we've got a number of studies looking at the impact of COVID-19 on the heart, the lungs, the brain, and the placenta, studies which I won't go into, but it's really showcasing how we can use these novel techniques and apply them in pandemics and other spaces. So I'd love to show this slide because sort of spatial takes a village. That's sort of your takeaway from this. And we initially did upfront spatial transcriptomics, traditional, discovery, unbiased whole transcriptome. And then we'd go in and validate using spatial proteomics using multiple different platforms, as shown here. But what we found over the last year or so is that as the plex has increased on the protein end, that has become more discovery for us. And now we're running pure discovery studies with the spatial proteomics approaches. And how these sort of data sets come together is this computational biology or spatial informatics piece, which is not just bioinformatics or biostatistics, but it's also imaging and spatial imaging partners that we're working with to really integrate these data sets together. And so what the takeaway from this slide is that spatial takes a village. These are complex data sets where you need pathologists, immunologists, oncologists, scientists, technologists, all, speaking different languages, but trying to dissect your disease and really understand that cell by cell. So I'll now go into a number of studies where we've used these approaches across a number of solid cancers. So this is looking at, in lung cancer, we had 90 patients, 45 treated with immune checkpoint inhibitor therapy in the second line setting, 45 treated with standard of care platinum therapy, and we ran 2 assays on it, looking at the transcriptome and the targeted proteome. And we gated on a marker, cytokeratin, to identify the tumor. And we looked at the non-tumor regions by looking at the non-cytokeratin positive regions. And so for every patient core within these tissue microarrays, we had a tumor and a tumor microenvironment or stromal mask where we had the transcriptome and targeted proteome data. And so we could then compare the expression profiles in the tumor compartments at the protein and RNA level, and those that were enriched significantly are those that are in the blue, between the responders on the left versus the non-responders on the right. And we can do this in the tumor. We can also do this in the stroma. And what was really interesting was IL-2 and SPP1. SPP1 is osteopontin, were highly enriched for in the response group, specifically in the stroma. And when we look at the contribution of IL-2 stromally located, almost by itself identifies the patients that will respond to immune checkpoint inhibitor therapy. So it really showcases how you can go in an unbiased manner and do some discovery studies using really well annotated clinical cohorts. This works. We're doing a lot of validation at the moment on this, but it shows you how we can identify markers, beyond PD-L1, PD-1 currently. And so we took these same tissue microarrays and we stained it with a low-plex 6-plex on the PhenoImager HT platform, looking at pan-cytokeratin, FOXP3, CD8, PD-1, PD-L1, CD68. So we can look at the individual cells that express these markers. We can look at the composition of the tissue microarrays, but we can also start to look at metrics such as the co-expression of markers as you move into the tumor and as you move into the microenvironment across individual cores and across individual tissue microarrays, and then extrapolate that into the entire cohort. And so when we looked at the cellular neighborhoods, so a neighborhood is saying that my tumor cell might be next to a macrophage, the macrophage might be next to a FOXP3 expressing cell, for example. We could identify 9 different neighborhoods within each individual core, each individual patient sample, and then we could extrapolate that neighborhood into the entire cohort of 90 patients and ask the question, what was the enrichment of the neighborhoods between the patients that responded to therapy versus those that didn't? And we could see that there was a CD68, PD-1, FOXP3 positive cell interaction that was highly enriched for in the non-response group. And so it's really taking this neighborhood model and using that to extrapolate across the cohort and look for associations with response or resistance. Are there certain neighborhoods that are enriched for in patients that do well versus those that don't? We know that when you compare early stage cancers to late stage cancers, we initially thought it was a random process, but there does seem to be some sort of underlying sort of almost arrangement as you move through to advanced disease. So it's a space that you could theoretically target in the future, but I think we're really early days with these neighborhood analyses. So we took the same tissue microarray that we ran the 6-plex on, and we ran the cycler platform on this, looking at a 36-plex panel. So 36 markers with single cell resolution, and you can start to see our heat map where we've got all our cell annotations, and we can start to look at the composition of each of the individual cores from the individual patients across the response group and the non-response group for the individual cell types that contribute those individual cores. And then we can start to look at the associations of those with response and resistance. And what we could see was that in patients that were non-responsive to immune checkpoint inhibitor therapy, there was a higher proportion of these Tregs, specifically in the non-response group. And so we can take that same data set, and what you have at the top is the individual markers for each of those cells. But at the bottom, what you have are the neighborhoods. So we can now look at the tumor neighborhoods, the mixed tumor neighborhoods, the Ki67, CD44 positive tumor neighborhoods, HLA-DR positive tumor neighborhoods, immune-rich B cells, effector cells, granulocytes, lymphatics, and stroma. So we can take the individual markers, but now we can assign neighborhoods based on that, and we can ask the same question. What is the enrichment of the neighborhoods between the responder group and the non-responder group? And we could see that in patients that responded to therapy, there was this increased macrophage mixed tumor profile. There was increased effector CD4s and mixed tumor profiles, blood vessels, or mixed tumor and blood vessels, and CD8 positive T cells and HLA-DR positive tumor cells enriched for in responder group. The reasons for doing the neighborhood analyses is that this gives us insights into cell types that'll be associated or close to each other. And potentially, if we identify unique neighborhoods that are associated with response, we can then move those towards sort of low-plex IHC panels that standard pathology labs can look at. And so we took this same model and did this graph neural network on it, looking at the nearest neighbor analysis, and we could see that the proximity of Tregs to monocytes, HLA-DR positive tumor cells to macrophages, or Tregs to CD8 T cells was associated with response or resistance to IO therapy. So it's really taking that spatial data and really trying to understand how are these cell types associated. How is the proximity of these cell types associated with response or resistance to therapy? And so we then took so we know that when we look at the TCGA data, higher expression of metabolic markers generally associates with a poorer outcome. So we looked at a number of markers, G6PD, hexokinase 2, citrus synthase, and so on. And we started to profile these entire TMAs with enzymes that identify these metabolic pathways. So this is just a snapshot of one of those tissue microarrays where red is high, blue is low expression, and we can start to see very clearly, if we look at GLUT1, there is varying expression levels across that. So this is on the PhenoCycler Fusion platform now, where we're using these metabolic markers to profile these tissue microarrays. And it's giving us sort of new insights that we can pair with the sort of immune cell profiling of these tissues. And so we took that data set and we applied it to a clinical trial cohort where this is a non-small cell lung cancer, where we had the entire cohort and we did a sub-cohort analysis on the Fusion platform, the PhenoCycler Fusion platform. And then we also looked at the sort of lower plex panels as well. So this is just showing you the survival curves for those cohorts. And what we found was that when we looked at the RNA for these profiles using spatial transcriptomics, we identified a GPX1, GPX4 enriched tumor profile, which was associated with patients that did poorly to immune checkpoint inhibitor therapy. And this was evident in every contrast. So patients that had complete response, partial response, stable disease versus progressive disease, CR, PR versus PD, or CR versus PD, GPX4 and GPX1 was highly enriched for in the tumor compartments of these tumors that did really poorly. So there was this almost sort of ferroptosis-like pathway that was signaling from the tumor specifically in this. And what we wanted to do was to see whether we could marry this data with spatial proteomic profiling of the tumor microenvironment to localize the signal. And so we applied the 57-plex fusion platform. This is the PhenoCycler Fusion platform, where we're looking at about 3 to 4 million cells per patient sample. So this is a lung cancer tumor where we're looking at about 57 markers, but this is just showing you 6 of those markers. So we can see the macrophages, the tumor cells, the B cells, the tissue lymphoid structures, the CD3, CD4s, and CD8s. And what we can start to do is map the entire tumor at 57-plex across these individual pre-treatment tumors against which we have RECIST 1.1 data against response to therapy. So whether the patients had PDs, CRs, PRs, or SDs. And what we found was that in patients that had poorer response to immune checkpoint inhibitor therapies, there was specific increased expression for these enzymes that sort of signal metabolic activity, specifically G6PD, GLUT1, specifically in the tumor of these patient samples. But when we looked at patients that had complete responses, so did really well to immune checkpoint inhibitor therapies, this metabolic signal was really dull. And this is really interesting because we know that when you get FDG PET scans of your body and you're looking for, breast cancer or prostate cancer, we're looking for macro levels of glucose uptake. What this approach allows us to do is to look at the metabolic activity at that micro level. How can we map the glucose uptake in the tumors itself? And so we can see very clearly, based on these metabolic features alone, that we can start to identify the patients that did really poorly, just based on a metabolic screen of the tissues, with the caveat that it is a targeted panel, but we sort of know what markers we're looking for at this stage. And so we took that same tumor, this is in that patient that had a progressive phenotype, and we started to map these markers individually. So the red is high, blue is low. So this is looking at the tumor in the tissue. And I'll just now start to pan through the individual channels for each of these. And so when we look at CD8, the CD8s are on the periphery. They're not entering the tumor. CD68, same, it's sort of overlapping with where the CD8 cells are. It's not really infiltrating the tumor. GLUT1, almost a gradient-like expression across the whole tumor region. G6PD, again, a lower gradient, but it's again expressed in the tumor region specifically. CPT1A is it's a marker for fatty acid metabolism, and that's localizing to where the T cells are in this tumor. This tumor is not infiltrated by T cells, but we can start to integrate these maps to really understand these tumors. And so we can put this data set together and look at the multiple pathways that are involved here, the amino acid, the fatty acid, glucose metabolism, and so on. And this is in a patient that did poorly to immune checkpoint inhibitor therapy. So it really showcases how we can go beyond just immune cell profiling of the tumors and really start to understand how these tumors functional in those time points that these tissues were taken. And so I'll go on to this skin cancer sort of biodynamics program where we've run two studies. Sort of this, the first one's called the C-MIX study, which is a retrospective cohort of about 50 patients, cutaneous skin cancers. And now we're running a TUMIC study, which is multiple time points over the course of immunotherapy. We get tissue at each time point, at cure and at progression, we still get tissue. And this leads on to a clinical trial called the Desquimate trial. And so we can start to look at these tumors, and this is just a gallery of these tissues profiled on the PhenoCycler Fusion platform, where we're looking at different groups of patients that have different clinical endpoints. So patients that had IO-sensitive tumors, IO-resistant tumors, and sort of those that had metastatic lesions. And this is just looking at a number of markers: pan-CK, CD3, CD20, CD68, CD163. So the beauty of this data is we can start to look at these lymph node-like structures, these B-cell-like structures, and using those as almost geographical features to then map to. And then what we see in this tumor on the left here is we have this highly infiltrated, macrophage-infiltrated tumor regions on the periphery, but really highly active tumor regions in the middle. We can take this same sort of immune cell profile of the tumor and now look at the composition of them. So this is now looking at the different sort of neighborhoods and cell types within each of these individual tumors. And this is sort of your base sort of reading into this, and it's where most of the field is. But what's really neat is we can take that metabolic screen and now we can map that directly back onto the tissue. So we can look at markers of hypoxia, HIF1, HIF2, and so on, fatty acid metabolism, glucose uptake, amino acid metabolism, and so on. And what you see is in the areas where you had that macrophage infiltration of the tumor on the periphery of this one on the left, you have high levels of hypoxia. You can start to see the oxidative stress and the other types of pathways that are activated in these tumors. So this is where the functional data gets really interesting when you marry this with the tumor and immune cell profiles of these tumors. And you can start to, see on multiple omics levels the types of data sets that start to interact when you put these together. And so we can look at the different phenotypes and the metabolic profiles of these skin cancers. And this is just looking at the different markers as you go through GLUT1, hexokinase 1, citrate synthase, CPT1A, HIF1 alpha, and so on across the individual groups of patients across this study. And so this is now when you look at that same tumor. So on the left, we have this sort of tumor on the periphery here, the active tumor cells. So the tumor on the periphery is almost dying or necrotic. The tumor regions in the middle, in the purple, are still alive and growing quite aggressively. But this is sort of the takeaway for me, is that from your single cell studies, you end up with these t-SNE plots at the bottom right. And t-SNE plots are great to understand the types of cells that are present in your population, but you don't really know where those cells are in space. And what Spatial gives you is we can start to map these profiles back onto the tissue. And so what we have on the bottom left is that the multiplex IF image taken, where we've now cell assigned these, and we can map those tissues back on to the tissue, and we can start to see, the necrotic dying tissue on the periphery, the active tumor cells in the middle, the lymph nodes in the green, and then so on. And we can take this, and this is in a patient that had a pre-treatment sample in a patient that had a pathological complete response to therapy, and we can start to look at the individual channels across this whole tissue. And so this becomes really powerful because this is now looking at 1.4 million cells at 35 markers. And this becomes powerful because this panel starts to define what a pathological complete response looks like channel by channel, where red is high and blue is low. Theoretically, we can start to identify groups of markers that'll define PET-CRs. And our ultimate goal here is, can we use a panel of markers to define a PET-CR on diagnostic tissues? And so I think whilst we have to do the high-plex studies initially, once we have sufficient numbers, we'll be able to, pool a number of these markers together and start to define what a PET-CR looks like. By the same token, we can look at a patient that's done really poorly to immune checkpoint inhibitor therapies. And so the tumor here is then the pink on the top right. And what was really interesting in this tissue was when we look at the metabolic markers, they were again highly increased in the tumor, specifically hexokinase 1, HIF1 alpha, GLUT1, Ki67, and so on. And so by the same token, we can start to identify on diagnostic tissues or early specimens, surgical specimens, what a pathological complete response looks like, but also what does progressive disease look like? And can we start to identify minimal signatures that define these patient groups? And can we then, identify it early in the piece so that these become predictive biomarkers? So really moving these assays into the companion diagnostic space. I don't think we're there yet, but I think in the future, using these multi-omic data sets and defining what a PET-CR looks like and a progressive phenotype looks like, we can start to think about these really exciting companion diagnostic assays. And you don't need a 35 to 100 plex, you might just need a 5 or 6 plex to define that. And so I'll go into this really exciting head and neck case. This work's under review at the moment, which we ran with Akoya Biosciences on the PhenoCycler Fusion platform. And so head and neck cancer, this is in a patient who had what's called a partial response to immunotherapy. So the patient responded but then progressed. So the patient had inherent resistance in their tumor. Our sort of working hypothesis was that, okay, we've got this patient's tumor. We know the patient had a partial response to therapy. Could we identify regions within the tissue that might have been sensitive, might have been resistant? And so what you see on the right here is the H&E of this tissue. This is what a pathologist looks at pretty much all day, a dual color stain of the tissue. And so what we are able to now do using these really exciting technologies is map over 100 markers onto this tissue directly. And so this is just looking at 5 of these markers. And you can see straight away, as I toggle between these two, you can start to see the tumor architecture straight away. So the tumor is in the top right, the top middle, and the bottom. And what you have is, this is an HPV positive disease. So what you have in the middle is normal germinal centers. So we can go on to generate these really pretty multiplex IF images, but now you've got co-expression of 100 plus markers for every cell of this tumor. And so we can do cell assignment, or almost like a cell atlasing approach of this entire tissue. And what you see in this tissue are these red and yellow cells straight away. These red and yellow cells in the middle are normal tonsil or germinal centers. But what you have at the top right is a tertiary lymphoid structure specifically infiltrating that bit of the tumor at the top right. Those tertiary lymphoid structures are absent in the area adjacent to it on the left, and you've almost got immature TLSs or really small unstructured TLSs at the bottom of the tumor. And so we can break this tissue down into subtumors. So taking this one head and neck tumor and breaking it down into subtypes. So we have tumor region 1, 2, 3, and 4. And the tertiary lymphoid structures are identified in that tumor region 4. They're absent in tumor region 2, 1, and 3. There are sort of remnants of TLS-like structures in tumor region 3, but they're not structured and they're not large. They're not infiltrating the tumor. So these are sort of naive aggregates. And so we know that the presence of tertiary lymphoid structures sustains an immune responsive microenvironment. There's a lot of literature showing, there's a lot of work by Tullio Bruno and a number of labs across the world have really showcased the power of TLSs. And what was sort of the takeaway was that these structured TLSs are what matter for IO response. It's not sort of these naive or unstructured TLSs. So there's a lot of work being done in this space at the moment. So we took this tumor and broke it down into about 835,000 single cells. And I'll just go through now the immune cell profiles within this. So this is looking at the multiple different components within this one tumor. When we look at the M1 macrophages, which are generally considered immune responsive, they are all localized to this bit in the yellow. The M2 macrophages are in the green, blue, and purple region, which are generally considered immune suppressive. And so we can start to identify on two dimensions this partial response to IO therapy. The presence of TLSs in one area of the tumor, which matches up to an M1 macrophage phenotype, but also the absence of TLSs throughout the tumor, but in those regions, the M2 macrophage phenotype is present. We can take this a step further and start to look at markers that pathologists look at. CD68 in the tumor, you can say the tumor is highly infiltrated in the green. CD8, again, you could probably say that tumor regions 2, 4, and 1 have higher CD8 infiltration compared to region 3, but on general, this is a highly infiltrated tumor. Even tumor region 3 has CD8 positive T cells in the white. And so what the takeaway for us from this study was that when we looked at the metabolic profiles of the tumor microenvironment, we saw that G6PD, a marker of metabolic activity, was highly enriched for in tumor region 3. And so that's the area which lacks tertiary lymphoid structures. It has this M2 macrophage phenotype and is highly metabolically active. It had high expression for a number of hypoxia markers, whereas tumor regions 4, 2, and 1 were enriched for BAX, which is a pro-apoptotic marker, and it had this TLS-like structure and M1 macrophage phenotype. So you can start to see how this functional data really complements what we're seeing at an immune cell level and an immune cell composition level. And so we took this data and we developed what's called a spatial metabolic map of the tumor. So you can see the pentose phosphate pathway is enriched for in tumor region 3. You've got fatty acid metabolism on the periphery and DNA damage. And then you've got expression of MMP9 and a number of EMT-like markers on the periphery of tumor region 3. And we think that that's the likely leading edge of the tumor, and that's the area that's spreading. That's the area that's metabolically active, and that's sort of what's ultimately going to kill the patient. Whereas in tumor regions 4, 2, and 1, we can start to see that these are they have increased expression for pro-apoptotic pathways, and that's sort of your dead or dying or necrotic tissue. So you can start to see the power of whole slide profiling of the tumor microenvironment and why we need to start to do whole slide imaging. It does become a cost limitation, but I think the biases you introduce in coring and creating tissue microarrays or sampling them, you might miss a lot of the really interesting biology that's happening that we can't see. And so we'll go one step further, and this is now looking at the individual neighborhoods. So this is looking at the tumor in the blue, the vasculature in the darker blue, the interfollicular CD4 T cells, the follicular B cells, the immune-infiltrated tumor in the red, and then the squamous epithelium. So this is now using that sort of neighborhood maps to identify unique neighborhoods within this tumor. We can start to then look at the RNA content within the tumor, the same tumor. And so we were looking at about 74 different markers across lineage, metabolic markers, stress, and chemokines. And what we found was that in the areas at the top, we had high expression of chemokines CXCL9, CXCL10, which had higher levels of CD8 infiltration detectable at the protein level. Whereas in the bit in the bottom, we had higher expression levels of CCL22, which is sort of this go away signal. And so you can start to see how the RNA data starts to integrate with the protein data when you start to marry these data sets together. And so where we're going with these tissues is we're starting to go from the H&E, the spatial proteomics data, the spatial transcriptomics data, the neighborhood analyses, and starting to build this omic cloud and then use that data to really impute this information into the H&E to identify pathology interpretable features on the H&E. And that ultimately democratizes spatial for the field. We sort of need to do these higher plex assays, these multi-omic assays to really figure out what is the underlying signal that is dictating response and resistance to therapy. And so we've taken that same model and we've now started to apply it to multiple cohorts, local cohorts, and in collaboration with Yale. So this is looking at two groups of patients from the Royal Brisbane and the Pre-A Hospital, where we have whole tissue head and neck samples, where we're looking at whole tissues, pre-immunotherapy tissues from patients that had CRs, SDs, PDs, and PRs against IO therapy. And what we can see is we can start to do sort of tumor stromal comparisons, but also tumor front comparisons. What we find is that in patients that have PAT CRs, they have this increased B cell presence, almost like this lymphoid-like structure, TLS-like structures within the tumor. And this goes back to our initial head and neck work. And we're trying to do a lot more work in this space, trying to really dig down into the signatures that define these. We think we need to pivot to whole slide profiling. That does become quite challenging when you're comparing across slides, and then there are technical challenges. But I think you really need to see the whole tissue to be able to understand the underlying biology that dictates response and resistance to therapy. And so where we think this is going is spatial proteomics is likely to outperform other sort of clinical assessments, standard PD-L1 expression, RNA profiles, NGS, because we're really understanding the tumor cell by cell across the entire tissue. And that becomes a really, really powerful approach. And really building that multi-omic cloud of information and using that to leverage that into the H&E becomes really powerful. And with that, I'd like to thank all the collaborators, the lab that does a lot of this work, Akoya Biosciences, who are very close collaborators of ours, Abcam, who've collaborated with a number of antibodies on these projects, the patients that have provided their samples to these studies, and the funding agencies. And I welcome any questions. Thank you.
Excellent. Well, Arutha, thank you so much for a fantastic presentation. We've got a number of great questions already, so I'm really looking forward to the Q&A. But as a reminder to the audience, if you do have a question that you'd like to ask, you could submit it using the Ask a Question tab on the right-hand side of your screen. All right, so let's jump right in. So spatial proteomics is a really exciting field of research in the life sciences. But are we actually learning new biology from studying these patient cohorts that were previously not possible with bulk or single-cell approaches?
Yeah, great question. I think what spatial has done is, especially having FFPE compatibility, so formalin fixed paraffin embedded compatibility allows us to go back in time and look at archival cohorts. So you can go into pathology banks, hospitals, and look at cohorts that are 20, 30 years old. You can get incredibly well clinically annotated cohorts, and you can look at, patients might have done exceptionally well to a therapy versus a group that didn't do well. And you can start to understand why these tumors did well almost cell by cell using these cohorts. And I guess the field's really moved on from sort of bulk into single cell and now spatial. But the spatial proteomics, I think, is sort of it's putting all these data sets together single cell improved on what we were doing with the bulk data, but the spatial is really putting it into the context. And for us, the beauty of this approach has been we don't need to go prospectively collect cohorts at this stage. Yes, we will need to do that for the validation studies, but we can go back in time and go back and look at really well annotated retrospective cohorts and start to identify signatures that we can drill down into and then prospectively go validate them. So I think it's really moved the field forward, especially for the clinical space. A lot of our oncology communities are really excited by these technologies because it gives us new insights into individual patient tumors and why they respond and why some don't. And in the patients that don't respond to therapy, how can we better target them? How can we better identify biomarkers that'll, we can give combination therapies to?
Excellent. Yeah, definitely a really exciting field to be in right now. So where you mentioned the spatial resolution of these assays, which I must say are yielding some pretty fantastic results. So what distances or resolutions are you actually measuring here?
Yeah, so we're going down into single cells and subcellular. A couple of years ago, the spatial field was sort of multicellular readout, but using these technologies, we now have single cell resolution. You do all your cell segmentation into single cells. We aren't really trying to push the boundaries and go subcellular at this stage with the proteomics approaches because we think the single cell approach is probably where we need to be to align with what pathologists can interpret and understand. So I think getting that information. So, for example, the lung cancer and skin cancer studies, we're looking at 3 to 4 million cells per patient tissue sample, and we're analyzing those across multiple studies. So, drilling down into single cells, I think, is probably our upper limit because that's the level of information that becomes informative for pathologists.
All right, great point. We actually have a question here about pathology. Does the data lend itself well to digital pathology? Is it really informative for the pathology community? I think you touched on that a bit, but maybe you could elaborate.
Yeah, no, absolutely. So, I presented this work at Pathology Visions last year, and it was really interesting to see how all the AI companies are, they're aiding pathologists in diagnosing cancer. I think where spatial is going to be informative is and where the pathology community were really excited was they could see that spatial is giving us new insights into the tissue that pathologists cannot see based on an H&E. So is it telling us about a new therapeutic that might work? Is it telling us about a functional state in the tumor that can be targeted? So these are information that a pathologist cannot read off an H&E that spatial can aid in informing them about the tissue, and then it helps in the therapeutic decision making for the oncology community. So I think as long as these assays are complementary to what pathologists do and it's not reproducing what they do, we sort of we're able to play in the same sandpit. And I think that's the position that spatial's really taken, and it's moving the field forward towards a digital pathology almost revolution, because as we build these omics clouds or these multi-omic data clouds, we use that information to drill down to a single cell or a pixel level what we can see on the tissues directly and what is pathology interpretable. And once we do that, that becomes really powerful because we can now start to identify features on an H&E that a pathologist looks at to then identify therapies that might work and aid an oncologist in their therapeutic decision making. So I think spatial is going to, in the next couple of years, really develop new ways that we're starting to understand what therapies to give individual patients. And I think it lends itself into, can we identify patients responders versus non-responders? Can we predict those patients really early from diagnostic tissue, if possible?
Great answer. All right, so is it all about plexity, or are there other factors like throughput or imageable area?
Yeah, that's a great question. I think there is a plex war going on at the moment. I don't think it's all about plex. I mean, plexity is great in that everyone's pushing towards the upper limits of we want to get as much information as we can from these tissues. But I think with the proteomics field, having assays, between 6 to 100 plex that you can run routinely, robustly, reproducibly, in a high throughput manner on clinical cohorts in a timely manner, where you can go from sample to due data generation and analysis within a timely manner. Ultimately, these assays need to be sampled to read out within a couple of days if it's going to be clinically actionable. So I don't think it's all about plexity at the moment. I think plexity is part of the story because we're trying to glean as much information as we can from these tissues, but I don't think we're going to be running hundreds and thousands of markers. Yes, potentially for discovery studies, but for the clinical piece, it is going to be a lot less. If we look at the ASCO multiplex IF assays that were published last year, they were all 18-plex or below for clinically linked signatures. So it's telling us that for the clinically linked signatures, we probably need to be around the between the 6 to 15-plex space. That's probably where we'll end up. So I don't think it's all about plexity. Plexity is sort of where I think in the transcriptomic space, yes, that is everyone wants to get as much information as they can, but I don't think that's a high throughput, clinically oriented assay that'll move forward to the clinic. You sort of need to have a targeted panel that you're able to run on every sample in a timely manner.
Yeah, that's a really excellent point. So which data analysis and visualization platforms are being used here?
Yeah, so my lab sort of split. We use commercial and open source. So we use a lot of QPath. We use a number of analyses tools that Akoya Biosciences provides with testing MCM Micro and a number of sort of cloud-based solutions as well. So I think the field is very I think the tools are being developed and they're being put out there, and we're testing them almost daily, the different approaches. There was a paper that came out a couple of weeks ago talking about how you can use pixels to annotate your tissues and sort of annotate your cells, and rather than your cell type clustering. So we're using a number of approaches developed by a number of labs in the US, but we're trying to standardize that workflow. So it is a lot of published approaches at the moment, and trying to do that in a high throughput manner is really, really important for us.
I think that leads well into another question here. Somebody's written that they're new to the field, and there's obviously a lot of tools on the market. So how do you decide on the technology and approach?
Yeah, it's a good question. I think the first question you need to ask yourself is, do you need Spatial to answer the question you're asking? I think a lot of people are jumping into Spatial without having a spatially relevant question. If you do, if that's a yes, then trying to figure out whether it's discovery or translational. For us, we did the traditional, let's just go unbiased whole transcriptome profiling of tissues, identify, our top hits, and then go validate them at the protein level. The traditional sort of approach that the field's been using for decades. What we've done, though, in the last year is we've pivoted to proteomics-based discovery. When you have 50 to 100 markers that you can run on these samples, that is also discovery targeted, but it is discovery because you are starting to understand the tumor microenvironment with a molecular readout that is, one, pathology friendly, but two, it's probably a lot more robust compared to your RNA signatures. So we're trying to really drill down on the protein. So I think for a new user, figuring out whether you want to do discovery science or translational science is probably your first question. But then also looking at the technologies that have good data sets out there, highly published. I think that's important. A lot of peer review papers is important for the field, not just pretty pictures. I think having robust peer-reviewed papers is important to show the robustness of these assays. And one of the really exciting things for us working with Akoya Biosciences was that we could see that there were a number of organizations, the Jedi Council, and a few other of these organizations, Astropath, and assay standardization working groups around the world, where they were running the same sample across multiple labs, and they could get robust data across them. So having reproducibility, standardized assays that you can run across the world in multiple labs and be confident in what you're reading is really important. You shouldn't have much assay variability. Those sort of pre-analytical conditions are really important. So Those are sort of the things that we look for when we're looking and deciding on new technology, and again, adoption globally. I think that's also important.
Excellent. Yeah, great answer. Thanks for that. Our next question here: how can you distinguish M1 macrophages and M2 macrophages in the tissue, and what phenotype markers were used to identify the two different polarized macrophages?
Yeah, that's a good question. We try to stay away from M1, M2s. The immunologists always tell us it's not as simple as that. But we use CD68, CD163, and a few other markers to try to, at a sort of a gross level, distinguish between the two. It is sufficient for the moment, but we are trying to improve our calls in that in the M1, M2 sort of space. But yeah, that's ongoing work.
Fantastic. I think in a sort of similar vein, how do you differentiate between the normal germinal center's lymphoid structures and the tertiary lymphoid structures?
Yeah, I wish I had an answer. No, it's a really good question. For us, the first step is we get the H&E, we get that annotated by a pathologist. We sit and we go through those individual H&Es that are path annotated, and we get the pathologist to annotate, where are the normal germinal centers, where are the, lymphoid-like structures, where are the TLS-like structures, where is the tumor. And when we have the lymphoid-like aggregates near the periphery or within the tumor, we demarcate them as tumor-associated or TLSs. There's a lot of work that Julia Bruno's done in the field where they're using multiple markers, CD19, CD20, CD21, the structure of these TLSs. We're looking at the transcriptomic data, so comparing the RNA between germinal centers and TLSs at the moment. So it's sort of an integrated approach at the moment based on pathology readout, multiple markers, where in the tumor is it very far from where the tumor is. So we're using multiple ways to differentiate it. I don't think we have a clear cut approach at the moment, but it's sort of this confidence level before we call them. We were talking to some colleagues the other day and they were telling us on DAPI that they could call lymphoid-like aggregates. So I think if you have these B cell-like aggregates together, we can start to label them lymphoid, lymphoid-like, tertiary lymphoid-like structures, and then sort of, as you molecularly characterize them, we'll be able to define them better. But it's sort of early in the piece where there's a lot of uncertainty, but a lot of research is being done. Olivia Newton-John's doing a lot of work in this space, Jessica Duarte-Gama, Julia Bruno, who are probably the experts in this space.
All right, so another question. Can spatial multiomics data be used to measure cancer progression in patients?
Yeah, that's a great question. We've started to do sort of primary tissue versus lymph node metastases versus distant metastases studies. And so, we know that primary tumors are very different to metastatic tumors in that the metastatic tumors are, they might be immune cold and then so on. How it can be used to measure progression is an interesting question. You almost sort of need to have, pre-cancerous lesions all the way through to stage 4. We are doing some early work in head and neck cancer and oral cancers, looking at, pre-lesion stage 1, 2, 3, and 4. I think that will give us insights into how, the pseudo-time sort of progression of these tumors, the temporospatial differences in these tumors. But it's sort of early days. You sort of need to be sampling across, tumor met. Sometimes the metastases isn't biopsied. So I think we need to look at, tumors which are more accessible, such as skin cancers and so on, and then start to characterize the primary versus the metastases or at progression. We're sort of early in the piece where we are looking at early data where it is diagnostic tissue or surgical resections. But I think over time, those sort of really interesting studies where you've looked at primary versus metastatic lesions will become mature.
Right, great answer. Another nice question here. When you look at responders and non-responders for ICI patients, have you seen any differences for differences in immune cell infiltration between them? So in responders or non-responders themselves, do do you see any different regions of infiltrations?
Yeah, we do. So, I mean, each study is sort of unique in that we're identifying different immune cell types that might be infiltrating, different neighborhoods which might be infiltrating. But it's also the state of those immune cells. So it's not like one cell type by itself will define it. And we can see from a lot of David Rim's work and a few labs around the world that there are these really interesting macrophage-like cell types that are found in the tumor. So it's not just about CD8s that are infiltrating the tumor. We are seeing clear differences between patients that respond versus those that don't. We don't think a single cell type is probably the answer. We think the neighborhood analyses and these cells that almost come together or pull each other into the tumor are probably where a lot more of the complex biology is, which are underpinning response or resistance. But where it gets interesting is when we marry these cell type information data sets with the chemokine and cytokine signaling. So how do these gradients of expression look like around those immune cells within the tumor and then so on? So trying to marry the RNA data with the cellular localization data will probably give us new insights into the responders and the non-responder groups. But yeah, it is sort of early days where the numbers are getting higher and the data is getting more robust. So stay tuned, I guess.
Yeah, definitely. All right. Well, I think we can cap this up with a question looking forward then. So Arutha, what are you most excited about? And then also, what keeps you up at night?
It's a great question. I think I'm most excited about how we can use spatial to democratize this approach for the field. So I think spatial is a tool, and if we use it correctly and we build this multi-omics data cloud and impute that information into an H&E, we democratize spatial for the community, for the field, to pathologists, and that becomes widely adopted. What keeps me up at night? I don't know. What does keep me up at night? I mean, for all our projects, we work very closely with patients and patient advocates and patient advocate mentors. I think ultimately these assays need to move forward to the clinic. It's great to have these amazing images and these incredible data sets, but hopefully this moves the needle forward for precision medicine, for personalized medicine, and we can start to treat individual patients' tumors individually, trying to identify, can we identify these patients that will respond to therapy early? If we know that these patients won't respond to immune checkpoint inhibitor therapies, is there another target we can identify? And is there a therapy for that? Is there an opportunity to give a combination therapy with immunotherapy in a way that is not toxic to the patient? So I think it's really, can we use these really exciting data sets to individualize therapies for individual patients so we treat their individual tumors and ultimately improve their quality of life?
Brilliant. Yeah, such an interesting field with so many developments to look forward to. Yeah, Arutha, thanks so much for joining us today and sharing your insights. It's really been a pleasure having you with us.
Thank you.
Definitely. Yeah, big thanks also to the audience here for participating. And last but not least, we'd like to thank the sponsor, Akoya Biosciences. And in closing, we hope you enjoyed this Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.